Hi, in this last video on RNA, we'll talk about how engineers might use tools to influence the RNA expression, which you might hope would produce a different outcome in the cell you're interested in studying. We'll talk about two methods, Antisense, which is about 35 years old, and RNAi, that's short for RNA interference, which has really revolutionized this whole field. It's about a technique that's 10 to 15 years old. We won't have time to talk about some new methods like riboswitches, which are done, for instance, in the Collins lab here at BU. But this is a rapidly evolving field, and there may well be many new methods in just a few years. So we've learned how to manipulate genomes at the DNA level using recombinant DNA so we can influence the outcomes of cellular behavior that way. Now we're focusing on two methods, again, antisense and RNAi, that's going to influence the RNA, that's going to interfere with the RNA's ability to make protein. Most drugs that are given to people influence the action of proteins, and it's, as you are well aware, it's a laborious process to identify molecules that will interact specifically with proteins. With RNAs, because of their base pairing capability, you at least have the principle of having exquisite specificity, directing some molecules to specifically bind a particular sequence and interfere with just that gene's function. At least that was the hope anyway when these methods were started. Using antisense technology, what we're going to do is we're going to develop short oligonucleotides that are complementary to the target DNA. We're going to modify them chemically somewhat, the DNA backbone, so that they're taken up better by the cells, that they have greater stability. And our only intention is for our synthetic oligonucleotide to bind the mRNA. We don't need the cell to do anything special with it. And as a result, since it's just a binding phenomenon, that this is not a complete knockout of gene expression, but it's more of a knockdown, meaning we can reduce levels 90%. If you want to get expression down to 0, you might use recombinant DNA and just cut the gene out or something like that. But nevertheless, the attractiveness of being able to target any gene is what really fueled the development of this technique. So here in red, we have a target gene. We have a ribosome that is going down the mRNA 3 bases at a time, 1 codon at a time, producing a polypeptide chain of amino acids. And the idea with antisense is to bind to the RNA and just cause translational arrest, provide an obstacle. When the ribosome hits this, it doesn't know how to interpret double-stranded RNA, and it will stop, or an RNA-DNA hybrid. And so that you haven't made a full protein, and thus the cell's behavior will be altered without this protein present, and you have created a change in the organism. Now, this is, again, a very attractive technology because you can target any gene. The practical difficulties of getting enough of the oligonucleotides into all the cells that you would need to block this to treat something medically has been a problem, and the stability of the oligonucleotide once is in there has caused some problems. Nevertheless, it was developed, and the discovery of RNAi was based on a proposed antisense experiment. Now, this is C. elegans. This is a worm that's about 1 millimeter long. You can just barely see it with your eye, but it's studied greatly in biology. And Su Guo wanted to knock down a gene called PAR1 and study the effects. How did the worms behave when this protein wasn't being made? So she's doing her experiment. You have to actually inject the material into these worms, and you have to do this on hundreds or even more worms. And so you can see part of the difficulty in drug delivery with antisense. But she got an odd result. She did an antisense probe, which she thought would be active. And then as a control experiment, she had put the sense. So as you know, this should not bind the target DNA anyway. Here's the target mRNA, poly-A tail. An antisense would bind to the RNA. However, a sense nucleotide would have the same sequence, so it's not complementary, so you wouldn't expect to bind. So this was actually her proposed negative control. But she observed something quite surprising. She found that the proposed negative control had just as big an effect on the organism's ability to survive as the antisense gene. So this was very curious. She thought she was inhibiting this gene. If this had been 0, would have suspected nothing. But the antisense and the sense gave the same result. And Sue was working in Andy Fire's lab at the time, and when they looked at the data, there were two things that they couldn't understand. First, the sense and the antisense RNA was producing effects, and secondly, with antisense, you have a stoichiometry, right? You can't inhibit more molecules of mRNA than molecules that you put into the gene. However, they were calculating the effects and how long they were lasting. They noticed the interference effects were even persisting into the next generation. And even a back of the envelope calculation revealed that they were getting a much larger effect than they could reasonably anticipate. This is a good drawing of how a sense oligonucleotide would not be expected to bind and produce any effect. And then they had a great insight into the problem. They thought maybe the sense and antisense oligonucleotides that we're putting into the cell might not be single-stranded after all. They were making their RNA antisense molecules using an RNA polymerase on a construct they had created, and it is known to produce random transcripts. So they guessed that perhaps the interfering RNA populations might be double-stranded. So then they did the next experiment, and in this experiment they were targeting another gene, and they used different synthetic methods to produce an antisense oligo and a sense oligo. And in both of these cases they actually saw 0 effect. So the typical antisense mechanism was not producing effect. But then they combined these two pure oligos to produce double-stranded RNA, put that in the cell, and now you have 100% phenotypic effect in the worm. Every worm that received this double-stranded RNA was getting a large phenotypic consequence. And again, here's sort of a calculation. They reasoned that they were injecting as little as 60,000 double-stranded molecules into each adult, and yet even 100 progeny from that adult would show this effect. So there could only be a few molecules per cell, and we know that there can be on average about 1,000 copies of mRNA per the cell. So how could so few molecules produce this effect? And they thought that perhaps it's not like antisense simply a binding event, but some sort of catalytic event. And so they constructed an experiment to test that hypothesis, and they were going to inject the double-stranded RNA that inactivates a targeted gene, and they were going to say, well, what happens to the mRNA of the targeted gene? In antisense, nothing really happens to the mRNA. It's sitting there. It's just blocked from producing a protein by the antisense. To test that, they were going to basically do a northern blot or in situ hybridization, take the mRNA, and with a probe molecule look to see if they could find the mRNA of the targeted gene. And what they saw was that whenever they injected double-stranded RNA, that the RNA is gone, and so they concluded that a different mechanism is occurring here, that somehow the double-stranded RNA that you are putting into the cell is degrading the target mRNA, not transcriptional arrest as in the antisense mechanism. So over the next 5 to 10 years in Andy Fire and Craig Mello at UMass, his laboratory, the pathway and the mechanism, what's going on with RNAi, was fully elucidated. Initially, long double-stranded RNA molecules were being introduced into the cell. What they realized, there's a protein called dicer, which will chop the double-stranded RNAs into small or short interfering RNAs, 20 to 22 base pairs long, that there's a protein complex called the RISC complex, which would bind the RNA duplexes then discard one of the strands and use this the strand that it retains as a template for the RNA to bind. Then when it binds the RNA based on complementarity to the retained strand, it's going to cleave the targeted mRNA. Once it's degraded, this active enzyme can go ahead and degrade another mRNA, and this is one reason why you can see such big effects from a small number of initial double-stranded RNA molecules. And furthermore, as you're degrading the RNA molecules, there is sometimes priming events creating more double-stranded RNA, which will get chopped up by dicer, and so you sort of have an enzymatic feedback loop producing more short interfering RNAs that will go ahead and degrade more mRNAs. So this is sort of an enzymatic amplification of the effect based on the original double-stranded RNAs that are introduced into the cell. As you might imagine, if you're starting to degrade entire long genes, that these strands might on average hit lots of other genes, and in fact in RNAi experiments you frequently can knock down the expression of a lot of genes somewhat, but typically your target gene the most of all. And then in a further development which made this technique very simple to use, rather than having to inject the C. elegans worms, for instance, with a needle or syringe to introduce the double-stranded RNA, they constructed bacteria with special plasmids where they'd cloned a gene of interest. They put T7 promoters. T7 is a promoter which is very constitutively active, so RNA polymerase will bind. It will make RNA from this strand, and since you're doing it in both directions, it'll make RNA from this strand. So inside the cell, you're actually making the double-stranded RNAs, and C. elegans eats these bacteria, and as you can see here, they are eating these worms that have been modified to express the protein GFP. They eat the bacteria that is going to do RNAi on the GFP gene, and as you can see, there's very little fluorescence. There's just a teensy bit down here. And so now it's a simple matter of all bacteria for all genes in C. elegans, for instance, have been made, and so now it's just a simple matter of feeding the C. elegans worms to knock down any of the approximately 15 to 20,000 genes that the worm possesses. Several years later, the techniques were worked out in other labs to do these on human cells, and it's not quite as convenient, but it's still been a very powerful method for confirming hypotheses about what happens when a gene gets knocked down. So here we'll compare some of the properties of antisense versus RNA. In antisense, we're putting in a single-stranded DNA oligonucleotide, and in RNAi, either large double-stranded RNAs, which will get converted to short interfering RNAs, or you can even just put in double-stranded short interfering RNAs directly. Here, all you're expecting is a binding event. You don't need any processing. RNAi only works in those organisms which have dicer and RISC in particular, or orthologs of those protein complexes, to perform the necessary events of chopping up large double-stranded RNAs into small RNAs, and then using the short interfering RNAs to target the mRNAs. There's a relatively low efficiency for antisense because it's not enzymatic. It's difficult to get the oligonucleotides into cells. Here in RNAi, this can be a highly efficient process because of, again, of enzymatic activity. Antisense in principle works in all species, and RNAi can only be done in certain species. However, they tend to be species that we're very interested in, and it's much more convenient than making genetically modified organisms. Okay, so that concludes our lectures on RNA analysis and technologies. Thank you for listening.